In this session, we will talk about the genetic code, or how the information in the DNA, A's, T's, C's and G's, is there to code for the proteins that we need to make. So we need to remember we've got DNA here. So DNA is in the cells of all organisms, and it is going to code for all the information needed to make all the proteins in the cell. So codes for all proteins. And it's going to be passed on from one generation to the next. The genetic code describes how this occurs, because we need to get from this DNA helix, oops, sort that out. We need to get from the DNA helix to the protein, and we need to code for the primary sequence of the protein. Remember, this is the amino acids that are joined together, and then once you've got the primary structure of the protein, then that will fold to the secondary structure, the tertiary structure, and even the quaternary structure for some proteins. But secondary, tertiary and quaternary are all dependent on the primary sequence, and the primary sequence is determined by the genetic code embedded within the DNA. Other piece of information, definitions to think about, are the differences between a gene and a genome. So a gene is the length of DNA that codes for a single protein. So one gene equals one protein. So length of DNA that codes for a single protein. The genome is the total set of genes in a cell. So the human genome are all the genes that make up, need to code for all the proteins in a human cell kept in the nucleus. That would be the genome there. So we know from previous work that we've got 4 different nucleotides of DNA. So we've got adenine, guanine, thymine, and cytosine nitrogen bases, and those nitrogen bases are attached in this position here. So 4 different bases, and those 4 different bases need to code for 20 amino acids. So that we need to use a 4-letter code to code for 20 different amino acids. And a problem scientists was working out how this could possibly work. So the way they were thinking, what you could do, you could say that one base equals one amino acid. Now this would only give you 4 possible combinations. So you could only code for 4 different amino acids. So that's not going to work. You can do some maths, so then you can say, what about 2 bases to code for 1 amino acid? Now we get more here. We get 4 times 4, or 4 to the 2, and that gives us 16 combinations. Now it's still not enough, because we've got 20 amino acids that we need to code for. So what about 3 bases to give an amino acid? Now this would be 4 to the power 3. So this is 64 combos. Now this is good. We've got at least 20, and we've got some extras. That's great. If you look at 4 bases, we're getting into crazy numbers of extras. So this would be 4 to the power 4, which is 256. So that's too much. We now know that 3 bases code for 1 amino acid. And we call these 3 bases a triplet or a codon. So either term is fine. Now, examples of what we mean by this. We know as biologists that GGT, read as a triplet, codes for the amino acid glycine. CTA codes for leucine. GCA codes for alanine. Now you don't need to remember these, but because scientists use a table to work it out, but you can use this table, and from a piece of DNA you can read off what the amino acid sequence, therefore what the primary sequence of the amino acid will be. So this is a wheel or a table, circular table, that you can use if you've got a piece of DNA to read off what amino acid you have. So we've got a codon. So if we have 3 positions, I am just going to pick some codes by chance. So I'm going to choose CTA completely by chance. So we've got the first base, the second base, and the third base. And what we would do with this system is the first position is read in this wheel. So you read the first position in the middle, then you read the second position, and then you read the third position. So if we're doing CTA, first position C, we're over here. Then we're reading T, so the next one is here. And then the final part is A, so we read across from A here. And then it tells us that CTA codes for the amino acid leucine. So we've got leucine, and that's the full letter name. Remember back to our work on protein structure, and each amino acid has a full name, a short name, and a 1-letter code name. And you can see all that information in the table. If we pick another example, just to go through it again, so I am going to pick AGG, so AG. Now, if we do this one in orange, oh, that's blue. If we do it in orange, so we had A, sorry about that, we have AGG, and we're going to look at it in orange. So A, we're over here. G, we're here, and then the final G means we're here, and it tells us that the amino acid in this case is arginine, also known as ARG, also known as R. So you can use the information to tell you this. This code is universal. So this means that you can use this codon table if you've extracted some DNA from a woolly mammoth found in the permafrost of Siberia. You can use this code to read off the protein sequence of that woolly mammoth. It's mostly universal. There are always exceptions in biology that make it more fun. There are some exceptions to things like the stop codons, which we'll come on to in just a minute. The code is described as being redundant. Redundant, or you can use the term degenerate. Now what this means is we said there was 20 amino acids, and using the codon system there's 64 combinations. So basically we've got some spare, some extra codons. So many amino acids are coded for by a number of different codons. So leucine, the example we happened to choose before, these all make leucine, and there's some more leucine over here. Okay, so basically if you want to be leucine, leucine has got 6 combinations. So leucine is I'm reading it the wrong way. Leucine is CTT, CTC, CTA, CTG, so I'm reading this one here, and then leucine is also TTG and TTA. So 6 different ways to code for leucine. So this is what we mean by degeneracy. The stop codons that we mentioned, there are sometimes some changes in this in some organisms. Stop codons don't code for an amino acid. So you've got a stop codon like shown like this in the table. There's 3 stop codons. Where's number 3? Okay, well we've got, number 3 is there as well. We've got 3 stop codons. So we've got TAG, TAA, and TGA. And these tell the protein machinery to stop. So they will stop translation when we talk about protein synthesis. So just like when we read written text, there are rules to reading the genetic code. So we always read the sequence in the same direction. So we're going to read that sequence 5' to 3'. That's going to be very important. We're only going to read one strand of DNA at a time. So when we're making the protein, we're only making the protein from one strand of the DNA. We read in a fixed reading frame. So what this means is we're going to start at the start codon and we stop at the stop codon. Excellent naming system. Start at the start, stop and the stop. Now the start codon is ATG. So if we look at that, so we're in A, T, and then G, like that. The start codon is ATG, and it codes for methionine. So we're going to start at the start codon. Stop codon, we said was here and here. We've got 3 different stop codons, and they tell what the protein machinery when to stop. They don't code for an amino acid. The code is also described as being non-overlapping. Now what we mean by this, we've got a stretch of DNA. This is gene 1, this is gene 2, and this is gene 3. Now you've got some extra bits of DNA in between that are involved in regulation, for example, but the genes don't overlap each other. So you would never get like gene 2 like here, and then gene 3 here. That would not happen. Instead, we absolutely have a gene followed by another gene, separated out like that. We said that when we're reading a piece of DNA, we're going to read it in codons, so groups of 3. And with any particular piece of DNA, there are 3 different ways to read it. So these are called the reading frames. So I'm going to take the sequence of DNA and write down the first reading frames. So if we start just by separating it out into 3s like this, we would have GAT, GCG, TAC, GTC, GAT, and then a T. So that's the first reading frame. And you would code that with the amino acids using the codon table, and you'd get the primary sequence of that protein. If we read it in the different reading frame, that would be how we do it. So we'd have a G, and then we'd read ATG, CGT, ACG, TCG, ATT. That's the second way you could read that sequence of DNA. The final way you could read it is if you divide it like this. So you would have TGC, GTA, CGT, CGA, like that. Now, these sequences of DNA, when they're translated into protein, would give 3 very different proteins, because these amino acids will code for will be coded for. And we'll do some activities of this in the seminar together. If we make all 3 proteins, then we will be wasting ATP and amino acids and resources on making 3 proteins when we only want 1 protein. And we would also have worries that we would be building up a toxic protein that we don't want. So instead we've got rules, and it's all determined, and the reading frame is determined by the position of the start codon. We said the start codon is ATG. And of these examples, the only one that has ATG is this one. So this is the correct protein. And this is the protein that will be made by the protein machinery in the cell, which we'll talk about in our next session. This will not be made, this will not be made, just this one here. So to summarize what we've looked at today, we've said that 3 bases code for 1 amino acid. We've said the code is universal, so you can read the code from a flea, a woolly mammoth, a human, all absolutely the same. The code is read 5' to 3'. We only work on 1 strand of the DNA. We start start codon. We stop at the stop. The code is non-overlapping, so you've got gaps between the different genes, and it's also important to remember that the code is degenerate. And having this degeneracy built in means that mutations or changes in the DNA sometimes can be okay, because you could just be swapping them for a different codon, but it would be coding the same amino acid. And we'll look at that more when we talk about mutation. Thank you.